I'm the program director for iTHRIV, and it's my pleasure to welcome
I'm ready
you
when
to
you are.
Great to today's edition of the Voices in Clinical and Translational Science seminar series. This series is an iTHRIV effort to
I can't
inspire
hear any
you.
of you now.
Sorry, hon, can you hear me? Yeah?
Yeah, we hear you, Sandra, but I'm afraid Margie might have a speaker issue now.
I got you now. I don't know, computer's being very weird today.
Well, it's October, so maybe it's getting tired.
Could be.
All right. Well, I'll tell you what, Mike, I'm going to turn it over to you to let you introduce our speaker for this series today.
Great, my pleasure. Thanks very much, Sandra. So it is really a pleasure to introduce my friend and colleague, Dr. Margie Lee. Dr. Lee is not only a great scientist and leader, she's a wonderful person. For those of us who've gotten to know her, I've had the pleasure of the last several years. She originally grew up in Bedford County, went to public schools there, and I think very early, right around the time of the Civil Rights Act, when schools were being integrated at that time. Very interesting time in history, and I'm sure for her personally. Margie did her undergraduate work in biology at Virginia Tech, and she then went on to do her veterinary degree at Virginia Tech as well. I think she was in like the third class of veterinary students or something in the very early days, and also one of the very first Black students to graduate from the veterinary school, along with one other colleague at the time, or one or two. She then went on from her veterinary degree to do a PhD in medical microbiology at the University of Georgia. She spent some time as a research associate up in the Midwest in St. Louis at WashU, but the call of the South was strong to her, so she she went back to the University of Georgia and joined the faculty there, became a professor of medical microbiology and parasitology. She also directed a center for food safety at the University of Georgia and the Population Health Science program, and was involved with the Diagnostic Lab there for poultry work, again at the University of Georgia. And so things come full circle in life. Sometimes about 5 or 6 years ago, Margie returned to Virginia Tech to take over the leadership of the Department of Biomedical Sciences and Pathobiology at the Virginia Maryland Regional College of Veterinary Medicine. And she's, being who she is, Dr. Lee, just couldn't resist taking on lots of other roles. I thought I wore a lot of hats, but she takes the cake, as they say, in that regard. She has served as the director of our Animal Cancer Care and Research Center here in Roanoke, at least as the interim director, and really helped that program. She's been a faculty member in small animal clinical sciences, and most recently, she was appointed as the associate dean for research and graduate studies at the College of Veterinary Medicine. So as I said earlier, Dr. Lee is very involved with research and involved in the clinical side of veterinary medicine and leadership. And in case you wonder, she can tell you more about where this drive came from and the interest in veterinary medicine. The story goes that there were a couple of ponies that appeared early in her life that I think were a gift to her dad or something like that. And she fell in love with the ponies and knew right then she wanted to be a vet, although apparently she had never met a vet before or seen one, but she just had it in her mind after meeting those ponies. So anyway, she went on and obviously accomplished that. She's done really great work in microbial pathogenesis, molecular ecology and epidemiology, and diagnostic microbiology as well. And I just want to save the best for last. When she has nothing to do, which I'm sure is less than 3 seconds a year, she hops on a Ducati motorcycle and rides all over the world. So without further ado, it's my pleasure to introduce Dr. Margie Lee to you.
Thank you for that. It made me sound more exciting than I think I am. I spent a lot of time thinking about, what to talk with you guys about, and how I think the trajectory of my career was kind of dictated by some of my choices and maybe some of my options. And I wanted to share with you, how that affected some of my choices. So I call this my adventures in snot and guts, because I think for many of us in microbiology, we have an unhealthy fascination with diarrhea and pus and snot. I am definitely one of those people. I will do molecular analysis of snot at the drop of a hat. So this is kind of how it rolls. The other part I wanted to share was really how you get there. So all of you guys, if I remember correctly on what I read and what I asked, have medical training, right? So your nurses, your physicians, your veterinarians, and you went into this, you thought, or at least I thought, with the desire to heal. And so I'm going to dispute Dr. Friedlander on one thing. I am not in the Department of Small Animal Medicine. I found out that I was not a healer. I'm more of a killer. So I ended up with poultry because in population-based science, we look at the sick ones to help people figure out how to keep the healthy ones healthy. So when I was a little kid, I grew up on a horse farm in Bedford County, and show horses were the big thing, racking horses, saddle horses, etc. And I wanted to be a horse vet, and I had the whole plan from the time I was 7 years old. And you guys will know what I mean when I say that, because doctors and nurses and veterinarians tend to know young before anybody can explain why or what we're going to do. And we stick with a plan for all that time in order to execute it. So I did. And when I was in high school, the news was that Virginia Tech would get the vet school for this part of the area. North Carolina State was building one as well, and I applied to Virginia Tech, early decision. At a time where it was a little sketchy, actually. You know, I'd been in integrated schools my whole life, but they had just integrated the schools in Bedford County when I was a little kid. And I wasn't really sure, honestly, if Virginia Tech was integrated, because even though on paper they were, there really weren't very many Black students here. When I was here as an undergraduate, there were maybe 100 out of, you know, close to, I think it was 17,000 at the time. I majored in biology, minored in chemistry. There's a story in and of itself right there. I was originally a religion minor, but that didn't work out. So I applied to vet school. I go to vet school. About halfway through vet school, I realized that equine medicine really wasn't going to be my thing, because I'm really analytical. I questioned everything. A colleague of mine, Rello, appreciate that. So Natalia, the head of small animal, and I were driving to a winery one day and decided to give each other a feedback on our strengths and weaknesses. And Natalia's weakness to me was, you question too many things. There's some things that you should just accept. And I'm like, I just can't. It's not in me. I question everything, which means I'm going to be in research, because we question everything. And so once I realized that I wasn't going to be a horse vet, I was on the fence on whether or not it was going to be dairy medicine, which is really analytical, or if it was going to be research. And I ended up going to the University of Georgia and getting a PhD and a master's in medical microbiology. And at that time, and you guys will be in some of these same positions, I was offered jobs. I had 2 or 3 faculty offers, a USDA scientist offer, all of which I turned down to go do a postdoc. And the reason I did is because I felt that in order to be able to do the quality of the work that I wanted to do and ask the questions that I wanted to ask, there were a couple of other things I needed in my toolbox. And molecular biology, but in particular, bacterial genetics was the thing. So I went to one of the premier labs. My postdoc mentor is now a member of the National Academy. I learned a lot from him about bacteriology, but I also learned a lot from him about translating basic science to applied science. He has a couple of patents on salmonella vaccines. 2 of them are actually in the market right now, and one of them is being used as the backbone for live vaccine development. And so in seeing how he developed that really taught me a lot about what could be done with just basic science. And, you know, I'm saying that because you understand. Here I am, a little brown girl from Bedford County, and I go to college, and I've got a PhD, and I'm a veterinarian. And my personality is such that I ask a lot of questions. How do you execute this, really? Because I have broad training, but deep training in one thing, but not in the other things. And so what I want to talk about is how you develop what you're doing with your career trajectory into something that can be impactful. So a veterinarian basically, we think of it as a healer of animals, but it's actually much more than that, just like a nurse and just like a medical doctor, where, epidemiology is basically the math of disease and disease control. Pathology is actually the mechanisms of disease. Toxicology, they're disease chemists. I mean, microbiology, we're trying to figure out what we can do with microbes or what they're doing with us. And so when I hit this crossroad of figuring out what I was going to do with this veterinary degree, there was something that was a little telling. And what I'm showing you here is the cover of a media manual. I worked in a micro lab as an undergrad, and my job was washing test tubes and glassware. But I was apparently fairly reliable, and I know I'm precise because all good researchers are a little OCD, especially if you're bench people. And so next thing I know, I'm making all the media for a project that they did at Oak Ridge National Laboratories at the nuclear facility. So all the water coming out of the cooling tower had to be cleared for pathogens before they could release it into the Clinch River. And so my boss, Dr. Klaus, who's retired from tech, had this project. I made the media, and I was such a little nerd at the time. Look at this media manual. I mean, so I've got this manual written up. Here's the purpose of the broth. Here's the background of how it works. Here's the chemical basis. Here are the ingredients and how you prepare it. And every single media I made had this kind of recipe. And so even as an undergraduate, I was analytical in how I approached things. So as a medical microbiologist, what I was looking at across the board in medicine, how could I apply this skill? So the obvious part that people do is study how pathogens cause disease. But there's a lot of other options. Come on, computer, here we go. Developing methods to detect pathogens, studying ecosystems and how microbes affect health. And what we usually thought about was that is the environmental part, right, of the disease triad. So the host, the pathogen, and environment. But what it's really developed into is microbiome analysis as well, environmental and internal. And then using microorganisms to do things. And that's what I got to see a lot in my postdoc, and then also with my colleagues at Georgia, where we were actually using the microbes to provide solutions, either for producing proteins, producing antibiotics. I had a colleague that worked on streptomyces, another one that mined organisms from deep sea trenches, and so hyperthermophiles to find enzymes that would be stable in industrial applications. And so you'll see how some of this marked what I did with my career. So I came back to Georgia and I was hired into the Department of Medical Microbiology, where I got my PhD. And I didn't come back because it was comfortable. I came back because Georgia had all the parts that I needed to be successful in research. So they had a microbiology department where there were basic science microbiologists. And then they had a number of departments where there were microbiologists, plant pathology, soil, food science, animal science. There was a USDA food safety group across the hall, and also this poultry medicine group. And so there were environments in which I could apply my microbiology skills to ask questions and find solutions. So I was in that department for about 12 years. I got promoted to full professor. And I'll throw this out because you might want to circle back around to ask me about these experiences later. But I was one of two first Black graduates from vet school here. I was the first Black PhD from their PhD program. I was the first woman when I came back to that department, female faculty member. I was the first African-American faculty member tenured in my college. And so there are this whole list of firsts that went on. So in that department, what I discovered was that as we had a change in department head, they were sort of trying to return to basic microbiology roots. And I wanted to do more microbiology in an applied translational way. And so I blackmailed the dean for a transfer to the Department of Avian Medicine. So I went over with the chicken people, the chicken and turkey people, because now I have an environment and I have a system for doing microbiology where I could do everything from basic science all the way to applied. And that's how I ended up being the director of the diagnostic lab. So I go in as a microbiologist, a veterinary microbiologist, working on genes basically, to finding a purpose for those things. But here's the real message I want you to take away from everything. Collaboration is going to be your big key. You can't be an expert in everything. You shouldn't try to be an expert in everything. I always go find an expert in something. I know how to do a lot of things. I mean, Mike brought up my motorcycle. Last week, I fixed my car. I blew a radiator hose. I managed to limp it back home. I ordered a part on eBay. I fixed my car myself. You know, I can do a lot of things. I'm a jack of all trades. But when you're doing research, you don't necessarily want broad in everything. You want really deep in some things in order to be able to get to the essence of the solution. And so the next research project I'm going to talk about will illustrate that. So this was one of the first big projects that I published out of my lab as a PI. And Pasteurella multocida is it's named after Louis Pasteur, in case you didn't know. And it's a big turkey pathogen. It's like the Black Plague of turkeys. 100 organisms introduced to turkeys and to rabbits, by the way, Rod knows this, can kill them in 48 to 72 hours. I mean, it is really, really virulent. And I worked on the organism as a graduate student. This is what I got my PhD in. And I remember then I was doing a study looking for virulence factors, just a general virulence factor screen. And this bug doesn't make anything interesting. I mean, other members of the family make an array of ADP ribosylating toxins that have just fascinating effects on hosts and explains the disease and the lesions. But those pathogens in Pasteurella's family are all host specific. So there's one that hits pigs and one that hits cows and one that hits sheep. And they're really host specific in the nature of the toxin. And multocida is called multocida because it'll kill anything. So in people, you guys may know, this is an infection that they'll see associated with dialysis because a couple of organisms exposed on catheters. It is the organism with cat bite abscesses and dog bite abscesses because it grows better in blood, straight, fresh blood, which almost no organism can do, better than it can grow in media in the laboratory. I mean, so the doubling time in blood is actually half what it is in the most rich media we can make for it in the laboratory. So in doing these screens, I detected an extracellular enzyme, a sialidase or neuraminidase. It's one of the enzymes that the influenza virus makes that allows it to attach and elude itself from cell receptors. Well, a bunch of bacteria make them, and nobody's really found a purpose for it. It was really interesting to me, and I remember writing in my lab notebook as a master's student, this is the only extracellular enzyme this organism makes. It must be important. I need to study this. So after going and doing my postdoc and learning how to do microbial genetics, I came back to study this. This enzyme, it turned out there are two, and just about every organism that has sialidase activity has two, and they have different enzymatic specificities. And I didn't know all this because this was really beginning the field of neuraminidase, sialidase research. And 4 labs I found I had attempted to study these in Pasteurella and had attempted to clone them and failed. And so here I am, an assistant professor The people on this paper, Marie was a technician, Adam's a technician, Amy was my master's student. But I hired a postdoc who was a biochemist because I knew I was going to be studying enzymes. And while I can direct the bacterial genetics part and the microbiology part, I wanted the enzyme part to be done really, really well. And so I found a way to get funding so that I could hire this biochemist. And in doing this, we cloned it. And I used kind of a novel approach. I didn't take the easy approach. I took the hard way because I said, what if this thing is toxic? It's probably not toxic in E. coli, but if it is, I'll never get it cloned. And so by doing that, what we did is we cloned both of them, we sequenced them, and then we started a whole study. It turns out, if you'll see here, here's the enzyme domain, here's the C-terminal domain. It ended up that there's a group of organisms that have an auto-transporter domain for their sialidases, and it ended up being really novel, and that's why nobody had been able to clone this thing. And if you guys have done any protein work, you'll find that there's some proteins really difficult. The sialidase domain has 24 anti-parallel beta sheets. The C-terminal domain has a bunch of them too. So there's something like almost 40 beta sheets of this protein. You can't even get it to migrate in an SDS page gel. And so it was like nothing about this project was easy. We managed to purify it, and it was really difficult to get it to migrate in the gel. You have to autoclave the protein because boiling wasn't enough to denature all those anti-parallel beta sheets. And then we looked at enzymatic specificity because we had two that were two different sizes. And it turns out what we discovered was that organisms that have sialidases tend to have two with different specificity that work under different conditions. And so for Multocida, which lives in the respiratory tract and in blood, this is how it gets food. And so we did growth studies that showed, here's the second one. Look at all these anti-parallel beta sheets. I mean, this is crazy. And so we did growth studies after making knockouts to show how it contributed to Pasteurellas' ability to grow in blood and in sialic acid containing compounds. And so this was actually a big deal for me because this was a project that other people who are much more experienced, the world's expert on sialidases, heard about my work through somebody else. And I get an email from him out of the blue, and he says, I hear you managed to clone this thing. That is amazing. And I'm thinking, wow, I mean, he knows who I am. But it was from being able to collaborate with, for instance, with Mizan, who was a really good enzymologist. I talked with another colleague. I got a cloning vector that was low copy and where you can control expression that enabled me to be able to do this when other much more experienced and better funded labs failed. And I was really proud. I got it published in the enzymology section of the Journal of Bacteriology, which is about as geeky as you can get in microbiology. So what did we do with it? Because I didn't set out, right, to be a sialidase enzymatic person, although I probably should have mentioned sialidases allow organisms to eat snot. So I need to circle back around to that. It is what allows them to live in mucosal surfaces that are covered with carbohydrate. This is how they get at the carbohydrate. So we published that paper. And then what we did, I had right about that time, a colleague's spouse walked in my lab and says, I want to work for you on Pasteurella multocida because we need a diagnostic test for rabbits. And I'm like, really? Who cares, right? Who cares about rabbits? And she says, rabbits are a food animal in a lot of countries. Multocida is the biggest pathogen they deal with. And there are 500,000 research rabbits in the US alone, of which this organism has to be eradicated from colonies. They will actually depopulate colonies because they're colonized with Pasteurella multocida, because even just colonization will interfere with results from experiments and studies that have nothing to do with microbiology. And there's lots of publications out there how it skews rabbit response to a lot of things, drugs, hormones, other kinds of infection studies. And so by using those molecular constructs for the sialidase, we used it as an antigenic target. And so we ended up collaborating with veterinarians who see rabbits clinically. They sent us samples, they sent us swabs. We ran them in our assays. We ran PCRs on the samples and confirmed that this was a really sensitive serological test for rabbits that were early colonized or persistently colonized, and then published that. And then because we're smart, right? Because clinicians don't necessarily read geeky microbiology journals, we collaborated then with clinical veterinarians to write a review, an application paper on how to use the test that was published in the Compendium on Continuing Education. So if you look at this, I took the geek part from the Journal of Bacteriology, applied it to something that could be useful, and then we wrote an article explaining to veterinarians how to actually use this. This test right now, and I'll show you a little bit about it, we patented the use of the proteins and we patented the coding sequences in case somebody actually wanted to sell the test as an actual commercial test. But what ended up happening is we had an infectious diseases lab, which was like an independent lab for diagnostics for exotics animals. And so we had a state diagnostic lab for veterinary medicine, and they didn't do many rabbits, and they didn't offer tests for oddball species. And so they licensed the technology, and this still runs right now in the infectious diseases lab. You can see it right here, Pasteurella multocity, antibody titer interpretation. I still get a little bit of royalty check on that. And you saw when the paper was published, when the test was developed. So Antech, and Rel will know this, Antech is one of the largest veterinary diagnostic companies in the US, and it may be in the world. Antech had a Pasteurella multocida serology test they were using. They ended up running paired samples with our test and their test. They suspended their test and they sent all their samples now to the infectious diseases lab and uses our test. So this is a really good example of how you need to be deep in something so you can figure out how to get at what you need in order to be able to use it for that thing you want. Don't be afraid to expand to application, and don't be afraid to embrace the really scary stuff. Enzymology was a section of biochemistry I hated. And we actually published a paper with enzymatic kinetics with an enzyme because I wanted what I could do with this protein more than I was afraid of enzymology. And that's what it takes sometimes. And you guys don't hesitate to interrupt me if you have a question. So again, collaboration is the key. Here's another example. I taught part of a National Science Foundation workshop in collaboration with a colleague on microbial pathogenesis. And in fact, the money that I got for the neuraminidase study, I got as a minority supplement on Duncan's NIH grant. He worked on mycoplasma, and there was some old literature that suggested that mycoplasma had sialidases. Well, I screened a bunch of his, they didn't. I screened a bunch of the veterinary ones, they did. So a colleague ended up getting a really impactful paper about diversity of mycoplasmas based on whether or not they had sialidases. But that summer that I taught that workshop, Michelle Furlong was my TA. And so when Michelle got ready to defend her master's, or no, actually it was her PhD, I go to her PhD, and it turns out, and I knew these guys, these were soil guys, and Barney Whitman is a microbial physiologist and taxonomist. So I go, and what Michelle did was 16S microbial ecology analysis of the intestine of earthworms. And I remember sitting in that seminar going, I don't care about earthworms. but this is really the kind of research that we need to do for animal health. So I talked with Barney, and Barney's like, oh, I've got all the primers. I will give you aliquots. We have got the software. If you want to come to the lab and run it so you can use our analysis tools, feel free. So here's somebody, he does taxonomy, like they name organisms. You know, there are those geeky people that like change the names of all the organisms. He's one of them. He's a member of the Berge Committee right now, which is the committee in the International Journal of Systematics that screws everything up for the rest of us. He also did a really interesting paper back when I was an assistant professor, and he kept calling me up and saying, Margie, how many organisms do you think are in a cow? And how many cows are there on the face of the earth? And after about the third phone call from Barney, I'm like, what are you doing? And he says, well, David Coleman and I are trying to estimate the number of bacteria on the face of the earth. Yeah, that kind of geeky. And they did, and they published, and it turns out something like 99% of the carbon on the face of the earth is actually tied up in microbes, and we live in a microbial soup. And they published this in PNAS as an estimate of microbial diversity, but it was fascinating. So I collaborate with this geek using 16S to characterize the intestinal community of chickens, because I'm in a poultry group, right? And I'm like, okay, chickens are the easiest to get to. So I get the microbial ecology direction from Barney. I hire a microbial ecologist, aquatic, to actually do the study as far as the analysis. The other people involved, Idris and I are both DVM PhD microbiologists. Barry's a pathologist. Chuck is a poultry medicine specialist, and John Maurer is another microbiologist. So rather than just going in and describing like what was in the intestine of birds, we tried to understand the ecology of how the intestinal microbial community is, which is what we called it at the time, develops in the bird and what we could use it for. And you'll see that this was published in 2003. So those primers in this study was in 2001. It was really right around 2000 when this kind of DNA technology became broadly available to environmental microbiologists, but very few were actually using it in vertebrate microbiomes at the time. So we were an early adopter in this. And what we found out was that there were successions in the development of the community that tied back to development in the bird. Like they hit these developmental thresholds where the microbiome would change, kind of tied to some sort of physiologic change in the bird. You know, and we report this information to veterinarians and to nutritionists. And it really allowed, which stunned me, them to be able to apply this information. So, and I'll get back to that. So we end up with about 24 publications on poultry microbiome. We looked at gene transfer, we look at development, we looked at the influence on pathogen colonization. So you guys are hearing a lot about C. diff and how fecal transplants can cure it. Well, we have known this in poultry since the 70s. This information is known, but what we didn't know was how it worked. So being able to do these studies, we found out how it worked. And you'll see that paper that I published has been cited almost 1,000 times. And so poultry researchers and poultry disease people were early adopters of microbiome research. So when I was doing this, and we were still early on in it, a group of veterinarians came over from Europe, and they were actually here for the International Poultry Expo. And I gave a short talk, and it exploded after that. I had no idea globally how poultry medicine specialists were interested in microbiome, and they began to use what they learned. And it was fascinating what I learned from them. So I give a talk, I'm trying to think, I think I was in the Netherlands. A veterinarian from Italy walked up to me and he says, this is what I observed. I can rear broilers on antibiotic free for about 4 cycles. The 5th cycle, they always get sick with some sort of gut disease. But if I swap houses and I put broilers in the house with the game fowl, because they raise game fowl for food too, and put the game fowl on the broiler litter, the broilers I can continue to rear antibiotic free for about 4 cycles before I swap them back. He says it doesn't make any difference with the game fowl, but it makes a big difference for the broilers. Well, what we realize is that for birds and insects, their microbiome comes from an environmental source. There's some in the egg and some on the egg, but it's how the organisms, their microbiome is actually seeded in the environment that allows them to get their normal flora seeded early. And so for us, the fecal transfer needs to happen day of hatch. You take feces from an adult bird that's healthy, you give it to baby birds, they're essentially resistant to all kinds of diseases, including clostridial diseases. So this information then went on in the industry to be used to develop antibiotic-free protocols that are for some companies like Tyson, which is the largest poultry company in the world, went antibiotic-free with some of these information. And so here's a great example. I go to an earthworm seminar. I start to ask questions within my field. I try to make it practical for the clinicians. They change the world and how they grow food just with this geeky information. Collaboration is key. So I think maybe I had been a full professor for about a year, and probably, what time is it? Make sure I'm not running over. Yeah, I'm doing good. Okay. So when I got tenure, I asked myself the question, what am I going to be? Like, what am I going to be? You know, I'm a microbiologist, but I'm not a microbial physiologist. I'm not a microbial geneticist. I'm a veterinarian, but I'm not a healer. I'm not a pathologist. What am I? And so I go to my colleague, Barry, and I go, Barry, what am I? You know, I'm just like a jack of all trades in research, but I'm deep in nothing. And he says to me, you're the guy who puts together the teams, which means you've got to be the guy that knows enough about each discipline. You've got to go deep in some to know how to put these teams together. And I really took that to heart. So I started putting these teams together. As I did my research, you saw one example there with the microbiome, and that was the team of people. And most of our papers that came out of that came out of the team. So I went and did a sabbatical, and that is really rare for veterinarians. I don't know how it is for MDs that are researchers. Basic researchers do sabbaticals at least every 10 years to try to stay fresh. So I got this crazy idea. I said, especially from the microbiome work, trying to understand how populations of organisms interact. So I decided to go do a sabbatical at Emory with a population biologist who was really a mathematician, which was kind of crazy. So I taught my veterinary bacteriology class in the morning. I jumped in my car. I drove to Atlanta. I worked in the lab on his project, and then I drove home. And if you guys have ever been to Atlanta, but if you've been to DC, you know what it is. It's a 2.5 to 3 hour commute back during rush hour. I'd go in the middle of the day at 62 miles. It'll take me about 60 minutes. I was losing a lot of time in the car. So I hired a student to go with me, and that student took dictation. And so the paper on the serological test, I wrote that in the car with the student. And it would be something like this. Okay, we're going to do 3 paragraphs as part of this section. Paragraph 1 will address blah, blah, blah. Paragraph 2 will address this, this, and this. Paragraph 3. Okay, paragraph 1 should be 4 sentences. Sentence 1 will be, and I would dictate, and we would get to the lab, and she would type, she'd look up references. I wrote a grant while I was on that sabbatical. But the crazy thing with the sabbatical, so I've got her essentially being my secretary. And keep in mind, students need jobs, right? So they want money too. So I found money to pay the student to ride in the car with me. And I drove and I would think. And learning from this population biologist how to think about, how to use computational sciences. I always thought the computational people used biological data to write the models. It's the other way around. You write the model, and then you go test the model in the lab, and you come back and change the model. And so Bruce Levin, who I worked with, wanted me to work on E. coli 0157, which I did a little bit, but Salmonella has always been the really interesting one. So I ended up, and the team on this is a undergraduate summer student, another microbiologist and I, where we were looking at whether or not the population biology of Salmonella control could work in vivo with bacteriophages, which you guys have heard a lot about phage therapy, right? And so if we could find a way to reduce Salmonella numbers in the intestine of birds, and Salmonella is so weird, it'll colonize chickens. You can kill baby chickens with 1 million Salmonella per chick on day of hatch. You give them 1 million Salmonella the next day when they're 24 hours old, you can't kill anything. It is an amazing microbiome question while you're considering what pathogen behavior is. And so because of that, if chicks get exposed and get colonized really easily, they are not sick. Can we use something to try to decrease Salmonella colonization? So what we did is we wrote a mathematical model predicting what the issues would be, and we set up an in vivo system with birds where we used the Salmonella, where we knew its genome, we knew its susceptibility to this particular phage, we had the genome of the phage, we knew the behavior of the phage, and all of this we could test in vitro. So in doing this, I wrote a mathematical model, and keep in mind, math to me was kind of like enzyme kinetics. It's like not your favorite thing, but all of a sudden now it is interesting to me because it may provide the answer to the question of whether or not this is even possible. And so I'll put it out there, the stuff that scares you the most, get a collaborator, learn how to do. What we found is that it is not possible to give a dose or multiple doses of phage to control Salmonella. So a colleague of mine took this information and applied it with a company that was developing bacteriophages. They found a way to encapsulate them so that they could deliver them every day to chicks, and ran a series of studies because we showed that the phage could not replicate above a threshold, and the absorption rate wasn't high enough where it could perpetuate itself. So he got around that by packaging the phage where they could deliver it and feed, and it controls Salmonella. Fascinating. And so recently I won a Poultry Research Award for some of this work, because even though I really like the geeky stuff, I really like applying the geeky stuff in a way that it can be used. So research accomplishments, diagnostics, we've done a ton of pathogenesis research, knocking out genes in Salmonella Pastorella, showing how it does things. Done a lot of antibiotic resistance research from a gene distribution standpoint. If you look at this paper right here, which is another one of those ones where I go to a seminar and I go, I wonder if that element's present then in these E. coli's we see, because they're always resistant to this, but we never use that antibiotic, and why is that? And what we find out is that there's a gene element perpetuated by the organisms in the environment that just kind of forces E. coli to be resistant. And so it's ended up in dozens of publications on how organisms become resistant depending on what kind of environments they're in. And I'll skip over that. And so as you do this stuff, you'll also be presented with opportunities for leadership. Okay, so advisory committees within your professional groups. Take those that are in your area of interest where you really think you can contribute, because you can really change how things are done, either in your discipline, in the government. I was on the national, NSAB, National Biosafety Advisory Committee for biosafety for the federal government. And we were the ones that came up with the DIRC and the gain of function research definitions. So when I'm hearing this stuff from the Senate and from the House, this was gain of function research. Like technically it really wasn't, because it doesn't work like that. We were the ones that did the definition. I've been on USDA advisory panels where we have come up with the regulations for pathogens in food and how things should be measured. You can really make an impact if you go to the trouble of actually learning how things work, not just the basic science, but how to translate it. And I think that might be my last slide. I'm going to skip over this one. I will say I'm in academia because I'm independent thinking. I can do what I want as long as I can get the money for it. It's like the best job in the world, but it's risky as heck. And I'm not really a risk taker. It's all calculated risk. Everything is calculated risk. But motorcycles are kind of my hobby. And that was 2022. I rode with a friend through Mexico for weeks and weeks and weeks, and I don't even speak Spanish. Perfect. You guys have lots of questions?
Thanks.
Wow.
Great story. I have a question. First of all, that was great. Thanks. Question about your anathema of kinetics and modeling, mathematical modeling. Looking back now in retrospect and looking at your journey and how all these things came together, what would be your advice to earlier career people who probably have the same, predilection of, biological sciences, et cetera, maybe not so much about the modeling, the math side of things, about, even though you don't see it right now for what they're working on a given project, but for taking a deep dive and arming themselves with those tools at this early stage of their career?
So the little note I made about sialidases as a master's student, I need to study these sialidases. And, in your gut, that's going to involve enzymology, right? There's just no way around it. You can play around with it. If you are willing to work from a conceptual basis, like if you've got this research project, this research problem, and as you develop hypotheses for that problem, it will be very clear to you at some point that either protein biology or modeling or carbohydrate chemistry is going to offer an insight into what hypothesis in this problem is likely to be true, right? So if you're willing to do the conceptual part, the other part starts to come if you're not afraid of it. And so for me, it was like, okay, the way to, if I'm looking at these sialidases, it's going to have to be enzymatic, but I'm going to have to knock them out. So I need to develop a genetic system. And I didn't give you all the background on that. On Piastrella multasa, there was no genetic system. So I had to make one and so I got 2 papers on that in order to be able to knock the enzymes out, in order to be able to study how they contributed to pathogenesis, in order to be able to clone the enzymes. So conceptually, when I said that, I want to study these enzymes, the path became clear. And that's when you do the deep dive, because that's how you're going to have to get at the answer.
Other questions? I guess anybody who wants to ask Dr. Lee a question, go ahead.
Thanks for that. I have to say, I thought I was going to be a horse vet as well. I was going to be a equine surgeon when I started vet school. So then I realized that was not for me. I was wondering how you kind of, when you talk a lot about building your teams, how do you coming from the clinical perspective, how do you get those basic scientists on your team and get them excited about what you're excited about? And how do you kind of build those relationships past, I'm pretty good at getting them excited, but moving past that to like, let's actually do something. So do you have any suggestions for that?
Yeah. So if you're going to be successful in science, especially as a team researcher, you got to be a good salesman, right? So I have to get people excited about chicken poop or salmonella. And, I'm not talking about sexy stuff. You know, can we do carbohydrate analysis on this mucus that comes out of the cecum? You know, you've got to find the right geeks for this kind of thing. And then you have to understand what's really fascinating about the system that you have to offer. You know, so I'm interested in these sialidases because I want to know what Pastorella was doing. But there are people out there that were interested in sialidases because they're like, what do sialidases do within the microbial world? And so I've had people seek me out because mine had novel features that allowed them then to hypothesize about the microbial world. And so finding those people that have a bigger view, pitching them what your system can do to advance what they're interested in. Okay, that's usually the sell. And then the second thing I do is I didn't just talk about it. What I tended to do is I'd go to their lab and run a couple of experiments, because why should I recreate the wheel if they're already running things? And then as we get a little bit of data, which means I had to make time to be able to do it myself. I could send somebody if I had somebody really good. But a lot of times I do it myself, and then they'd see the data and go, okay, this is really cool. And you kind of set the hook. The third thing you really have to do is you got to find a source of funding, right? Because you can't keep doing everything on a shoestring. So you've got to be really good at being able to go after money, small pots to start, to pay for getting data that makes it even more fascinating. But you really need to have a cogent plan in there. Okay, so if we're going to do this, then we're going to have to play for the carbohydrate analysis. I got money for that. And now that we've got the results, and if we have this, you got to have a plan. Does that make sense?
Yeah, that's really helpful. Thank you. I like the idea about like, getting people excited about your model, because I think that's what I can offer, you know, more than other, or that's different than other people, I guess.
Yeah, and I mean, you've got cool stuff, because I mean, what I always loved about clinicians is I could pass a clinician in the parking lot and I go, what's going on today? That's really cool. And they'd always have something. And I'd go, in fact, one of the most recent papers, I have Pastorella again, another diagnostic test. And a clinician says, you know, these companies have been using this live vaccine, and it's a serotype 3x4, but they're having disease in birds about 3 weeks after they vaccinate. It's a different serotype. And I'm not convinced that it's not vaccine-related disease, but it makes no sense. And I'm like, okay, I could geek out on that. And sure enough, we went and got genomes, we sequenced genomes, we made a vaccine-specific PCR. Turns out that one of the batches of the vaccine strain from a particular manufacturer had dissociated to a virulent strain. So I called up the rep and it's like, you need to see this data because you guys need to run this quality control test to be sure that you're not releasing batches that are going to kill birds. And published.
Hi, Dr. Lee. Thank you for the amazing presentation. I had a question about something that I've been kind of thinking about since I really started my research career that you brought up a few times, which is the idea of team science. And what I've been kind of struggling with is, as an early career investigator, I not only appreciate, but I find it very necessary to bring in and collaborate with other people who have areas of expertise that I don't have. And frankly, I'm not particularly interested in developing. But when you also are the PI on a paper and you're, you know, trying to be a good scientist and, you know, establish credibility, but do your science well, how do you kind of balance letting, say, the expert biostatistician do the biostats without understanding all of what goes into that? Or how do you kind of like let go of enough control to really be a team player in the team science, but also balance that with having to be responsible as the PI for all of the data and everything that is going into your manuscripts?
So that's the advantage of going to the collaborator's lab and seeing how things are done, right? Or in the case of the biostatistician, if they can't explain it to you in a way that makes sense, you know, because I have really high standards on what I publish and who I work with. And I really don't work with people that I think have lower standards, because at some point it bites you in the butt. You know, and the sialidase thing's another great idea. So I ended up finding out when I first sequenced it, we sequenced it into a transposon, and there had been a theory that sialidases had been horizontally transmitted by jumping genes. And so I sequenced into a transposon in my clone, and I'm like, and so the sialidase people are all excited because I've got the goods now, right? But I'm like, wait, okay, hang on a minute. Let me do a couple other experiments because this just seems way too easy and way too convenient, and I don't think I trust it, and it might bite me in the butt. And sure enough, it was a cloning artifact because E. coli hates the gene, and it jumped from the cloning strain genome into the clone in order to turn off that auto transporter part. And so if I didn't have such high standards, I would have fallen for that. And I would like to work for people that have that same sort of standard, right? So if you go to the labs or you go to their workplace and you see how they do things, you can get that assurance that their standards are as high as yours. And so I'm picky about who I work with. I really am. In fact, I've given data away where, I've done something for someone else's experiment and they want to put me on the paper because they want to have me on the paper. And I'm like, no, I don't really need that. Thank you. I'll just give you this data because no. So some stuff if you've got really good collaborators, you don't have to worry about it. You go read their work and you go see how they do it. And then you can let go of the control. I'd love to know what Mike says about that.
I'm not getting dragged into this one here.
Yeah.
Well, we are nearing the top of the hour, so I think we're gonna need to wrap things up. That's a great place to leave it for the day, though. But Dr. Lee, you have given us a lot to think about, but I most appreciate your comments about collaboration and building your research team and true team science, which is really just resonates with my heart for all of the work that we're trying to do in iTHRIV. So really appreciate your time today and all the wisdom that you have shared with us from your experience. So thank you very much, and appreciate everybody coming to our series today. Join us again next time, okay?
Thank you guys for the invite. I really enjoyed it.
Thanks,
Thank
Mark.
you.
It's great. Thank you.